When we talk about DNA mutations, we will specifically be talking about the mutations found within nucleic acids and nucleotides because those are the building blocks of our DNA. Now we're going to go over 5 of the most common mutations found within DNA or nucleotides, and the first one is deamination. Now if we break down this word, we can understand what it means. Amination, it has that A-M-I-N, amin. So it must be something related to an amino group. And then we have D. So D means remove. So this must be a removal of an amino group. Now, if we take a look over here, we have this cytosine, and cytosine is a nucleotide. It has a nucleotide with an amino group atop up here, this NH2 group. Now, if we remove this amino group, we end up with uracil, and uracil is a different nucleotide. It is not the same as cytosine, and uracil is actually not found within DNA. In DNA, we have A, G, C, and T. We have adenine, guanine, thymine, and cytosine. We do not have uracil. So we can imagine that through deamination, we get this different nucleotide, or we get a completely different compound that we do not want within our DNA, because that's going to result in some sort of a mutation. So the first mutation is deamination, which simply refers to the removal of an amino group. And these are just various other examples of mechanisms where we see that deamination. So over here we have this NH2 group, which is removed, so we get a completely different compound, and we see NH3 as a resulting product. So as this amino group is cleaved and it's departing, it picks up another hydrogen and it becomes NH3. And this is also just another example, similar to our cytosine example, where we remove the NH2 and we get this double bonded oxygen carbonyl up here instead. So first DNA mutation, deamination. Now the second type of mutation is depurination. So we can break down this word once again, where D means remove, and we have purination. So that refers to purines. Now remember, when we talk about nucleotides, we have 2 different types of nucleotides. We have purines and we have pyrimidines. Now here we're talking about purines. So that means we're referring to A and G, which is adenine and guanine. So removing of a purine, so we're removing either adenine or guanine. Now over here we can see in our example where we have a nucleotide, we have that pentose sugar, we've got that phosphate group, and we've got this guanine nucleotide, and it's undergoing this reaction where our guanine has been cleaved and separated, and we get this compound as a product. So depurination simply refers to the removal of a purine, either adenine or guanine. Now we can also imagine that if we remove a nucleotide from our DNA, that's also going to cause changes, because we're altering the original format of our DNA by removing a nucleotide. The third mutation that can occur in DNA is due to UV light, and we hear this all the time, to always wear sunscreen to protect against the damage against the sun. So here, what happens exactly, specifically in this example, is that whenever we have adjacent thymines within our DNA, and those adjacent thymines are exposed to UV light, we get 2 products. We get 2 photoproducts, one being cyclobutane and a thymine dimer, and the other is a 6,4 photoproduct. So once again, we can imagine that we are changing the original format of our DNA, and we're changing it to something completely different that isn't really a nucleotide. So we're going to get some mutations, and that's not beneficial towards our DNA. And what actually happens within our DNA when we get these 2 photoproducts from the UV light interacting with the adjacent thymines, we see this kink within our double helix. You can see over here that we had those two adjacent thymines that are stacked on top of one another, and we see this kink or this irregular bend in our DNA, which is obviously going to cause mutations because we're altering that original format of our DNA. So far we've gone over deamination, depurination, and now UV light can also cause mutations. So the third is chemical agents. So obviously some chemicals are going to be harmful to our DNA. This type of mutation is pretty straightforward. So we have these two kinds of compounds where we can classify them as nitrous acid precursors. So any nitrous acid precursors, they are going to cause chemical damage to our DNA. And then we have the alkylating agents, which are also going to cause chemical damage to our DNA. This is super straightforward. These two types of classified compounds will cause chemical damage. Now the last one is oxidative damage, and you've probably heard of this one already, but this is due to free radicals, which essentially means that there's only one electron in that all alone, and it's unpaired, and it's going to be highly reactive. So if it's highly reactive and it reacts with our DNA, that's going to cause some oxidative damage to our DNA, which is going to result in mutations because it will be altering that original format of the DNA. One last thing to point out where we were discussing UV light, I forgot to mention that X-rays and any other radiation, ionizing radiation, can also cause damage to our DNA, because just like UV light, any type of radiation, it will cause some damage to some extent. But other than that, those are the 5 main types of mutations, the 5 main causes of mutations. So depurination, deamination, UV light can cause damage, chemical agents, and then oxidative damage due to free radicals.